Before we can allow B and T cells out into the body, into the circulation where they may encounter a pathogen, they have to be tested. This testing is to ensure that those B and T cells do not go into the body and start attacking our cells, our self-antigens. So this testing, called selection, takes place in the bone marrow if we're talking about B cells, or in the thymus if we're talking about T cells. Either way, though, the testing is conducted by antigen-presenting cells, such as macrophages. Now recall that antigen-presenting cells always have an MHC2, which is like a wanted poster. And in the circulation and in the body, that MHC2 always displays foreign antigens. However, in the process of selection, these MHC2s are going to display self-antigens to the immunocompetent or immature B and T cells. Now, the test is fairly simple. The antigen-presenting cells, of which there are many, each displaying a different self-antigen, go and display that MHC2 with the self-antigen to each of these B and T cells, and there are millions of these. So this is a long testing process. And the test is fairly simple. It's 2 questions. The first one says, can you read the wanted poster? Do you recognize the MHC2? Do you, as a B or T cell, know that you need to look in this MHC2 to see if you can find the antigen that your receptor matches? So, if a cell cannot recognize the MHC2, it just comes up to the antigen-presenting cell and goes, duh, then the answer is no, I can't read the wanted sign or the MHC2. And any cell that can't even read that is kind of a dud. And while it's not killed, it is removed from the population that will eventually make it out into the body. In fact, they go dormant. So the antigen-presenting cells are like, we got a dud here, and they just get rid of it because it's not going to serve any purpose if it does go into circulation. If they answer yes, I can recognize the MHC2, then they get to go to the second question. The second question says, do you recognize and want to attack the antigen that is being displayed? And we want the answer to be no here, because these are all self-antigens that are being displayed by the antigen-presenting cells. However, a very large number of these cells answer yes, and those cells are killed by the antigen-presenting cells, because if they made it out into the body, they would recognize self-antigens and would initiate an attack. So, if the B or T cell, immunocompetent B and T cells answer yes here and no here, right? So these are the proper responses. Then they get to go to the next stage. If they answered no here or yes here, then they are eliminated from the population that's going to be allowed out into the body. Now, in terms of how many cells actually make it, there's about a 2% success rate. So 98% of the cells that go through this testing fail and are not allowed out into the circulation. Only 2% recognize the MHC2 and say, that's not my antigen. And then they are allowed on to the next step.